Hi guys, Ernest here. In our video today, we're going to look at the concept of a under DNA and RNA, and this is the DNA double helix bond, double helix structure and the hydrogen bonding. Now we said that the DNA basically is formed by this particular formation of a phosphate molecule, a sugar, and a nucleic base, and we're going to see that this particular formation alternate in a way that these specific bases are bonded to the other bases of the next, or rather the alternating, the strand that they are alternating with using this specific kind of bonding. Now, basically, this is what we are referring to as a bond between the atoms of the two bases for the guanine and cytosine that I've illustrated using the dotted lines are the ones that I'm referring to as the hydrogen bonds. And shortly, we're going to see how these specific bonds are occurring using the figurative illustration on our right. Now, in this case, if we have a phosphate molecule and then this is a sugar and then a base, and this concept is going to alternate in this specific order. Now, bearing in mind that guanine has got a particular site for a receptor for the cytosine molecule, thymine is going to have a site for the receptor for adenine, cytosine for guanine, and adenine for thymine, and this is for the DNA structure. And if in case we have an RNA structure, in this case, in case we have an RNA structure, in this case, then we said that thymine is going to be replaced by uracil. And basically for this specific set for the right, left side of our body is illustrating the double helix structure of our DNA. Now, basically a section of it, because as we said, this specific has two strands intertwine and they form a double helix as shown in this particular case. Now, the point of attachments, we said that is this carbon number 3 and 5, and the bases are attached at carbon number 1 of this specific sugar respectively. Now, on our right side, this is the base of guanine that is being attached to cytosine, as we said that this is the one that has a receptor site for it. And at this specific point, we have what we call hydrogen bonds. Now, for adenine, for guanine, and for cytosine and guanine, they are going to have this respective hydrogen bonds, which are 3 in number. For oxygen and hydrogen, hydrogen and nitrogen, oxygen and nitrogen, hydrogen and oxygen, respectively. Now, for adenine and thymine, they are going to have 2 hydrogen bonds. And then talking about the concept of hydrogen bonding, in our introduction to our organic chemistry, we have talked in one of our videos about the concept of bonding, especially now that we said that elements in the periodic table on both extremes will prefer either to lose or gain electrons in order to form stable compounds. But those ones which are found on the middle, or rather relatively on the middle of the periodic table, would rather like to share the lone pair of electrons in order to attain stability. Now, in this case, we came up with the concept of covalent bonding. And for this case, for hydrogen bonding, we're going to see that these are the type of bonds where an attraction between 2 atoms that have already participated in bonding, as we can see. For instance, hydrogen and nitrogen are bonding, but they have already participated in bonding of this respective structure of their individual molecule for guanine and cytosine. And therefore, for the definition of a hydrogen bond at this specific point, it's an attachment for hydrogen atom and another electronegative. We have already talked about the concept of electronegativity in one of our videos. So in this case, if this hydrogen is attached to this one, this is an electronegative atom that is found on this side, whereas the cytosine is going to form this specific hydrogen bonding. The kind of the organized kind of arrangement for the DNA molecule and how we can be able to expand these specific points of attachment in order to show the hydrogen bonding. Now, I hope that in a discussion about the concept of DNA, we have already looked at what it's made up of, specific sites at which these attachments, the individual bases, can be able to be put. For instance, for this, as I've said, they are located at carbon number 1 for the 2 strands, And then the point of attachment for the phosphodiester bonds is at point number 3 and number 5 for the 2 strands respectively that move in those respective direction for the concept of interweaving, as I have shown in this particular case. Now in our next maybe other discussions, we're going to use this double helix structure. Now that we have expanded more on the chemical properties, or rather the chemical structures of these specific molecules, and already discussed them in details on our various videos, I'm going to put a link on this discussion in the description box, and we'll try to see how these specific molecules can be able to be treated in the concept of genetics and inheritance. So that's it for the discussion for this specific case. Now for us, for this particular video, we were just to try and mention and show the hydrogen bondings that I've already illustrated, and show how the positions of these specific bases that we have mentioned and the sugar phosphates are being attached, and the specific hydrogen bonds that I've already illustrated at these specific points on our discussion. So that is it for today, guys. Bye. Let's meet on our next video. God bless you.